The signal sequences are typically residues long rich in charged residues such as arginines as well as hydroxyl residues such as serines and thronines but devoid of charged residues. A28 to 80 positively negatively. B300 to 800 negatively positively. C28 to 80 negatively positively. D300 to 500 positively negatively. The correct answer is 28 to 80 positively negatively. Which of the following is an incorrect statement about SignalP? A. It only uses neural networks. B. It only uses harms. C. It is a web-based program that predicts subcellular localization signals. D. It uses both neural networks and harms. The correct answer is it only uses harms. Chloroplast localization signals are also located in the terminus and are about 25 to 100 residues in length containing very few charged residues but many hydroxyl tail residues such as serine. A. N negatively. B. C negatively. C. C positively. D. N positively. The correct answer is N negatively. Which of the following is not one of the training sets in SignalP? A. Prokaryotes. B. Eukaryotes. C. Gram-positive bacteria. D. Gram-negative bacteria. The correct answer is prokaryotes. Which of the following is an incorrect statement about the terminologies related to protein sorting? A. Subcellular localization is an integral part of protein functionality. B. Many proteins exhibit functions only after being transported to certain compartments of the cell. C. All the proteins exhibit functions after being transported to certain compartments of the cell. D. Protein sorting is also known as protein targeting. The correct answer is all the proteins exhibit functions after being transported to certain compartments of the cell. Chloroplast localization signals consist of two adjacent signal peptides, one for targeting the proteins to the stromaportion of the chloroplast before being cleaved and the other for targeting the remaining portion of the proteins to the thylakoids. A. True. B. False. C. D. The correct answer is true. For many eukaryotic proteins, newly synthesized protein precursors have to be transported to specific membrane-bound compartments and be proteolytically processed to become functional. A. True. B. False. C. D. The correct answer is true. Once the proteins are translocated within the organelles, protease cleavage takes place to remove the signal sequences and generate mature proteins. A. True. B. False. C. D. The correct answer is true. TargetP is a neural network-based program similar to SignalP. A. True. B. False. C. D. The correct answer is true. The signal sequences have a consensus but contain some specific features. They all have a core region preceded by one or more positively charged residues. A. Weak hydrophilic. B. Weak hydrophobic. C. Strong hydrophilic. D. Strong hydrophilic. The correct answer is weak hydrophobic.